Welcome to the pre-harvest fruit drop section of the Flowering and Fruiting short course. This video will focus on the causes and mechanisms behind mature fruit drop, or pre-harvest fruit drop, and how HLB is affecting pre-harvest fruit drop in Florida's current climate. The objective of this course is to understand the physiology of pre-harvest fruit drop abscission, or in other terms pre-harvest fruit drop, explore the roles of phytohormones in abscission, go over some of the environmental roles in abscission, and lastly talk about how HLB is affecting pre-harvest fruit drop in groves. To begin, the term fruit drop is also referred to as abscission. Abscission is a natural physiological process by which plants shed plant parts and organs. Abscission is a highly beneficial process that allows the plant to discard diseased or damaged organs and also to allow for the dispersal of seeds for reproduction. For this course, we want to differentiate between a fruit that might break at the stem due to forces from winds or grove equipment, which is shown by the fruit on the left, and fruits that detach naturally from the tree or go through the abscission process, which is shown on the right. Growers may see more fruit on the ground after inclement weather, but it may not be due to fruit drop but from forceful removal from the stem. Abscission occurs in distinct areas from the plant, referred to as abscission zones. There are two types of abscission zones in citrus. Abscission zone A, or AZA, occurs at the top of the peduncle on the stem, and abscission zone C, or AZC, occurs at the calyx at the base of the peduncle where the fruit is attached to the stem. The two different zones allow for multiple types of fruit abscission to occur, such as fruitlet abscission, which happens at AZA, and the natural process of the yearly June drop, which occurs at AZC. This video will focus on the pre-harvest fruit drop, which occurs at AZC during stage 3 of fruit development, which is when the fruit generally stops growing and is in the maturation process. This type of fruit shedding is prior to harvest, hence the name pre-harvest fruit drop, that results in profit loss for growers, so understanding why and how it's happening is very important. Currently, the mechanisms behind pre-harvest fruit drop are not fully understood, but there is a lot of research behind this natural process that can elucidate some of the mystery. There are multiple physiological stages of abscission that can be summarized into 3 steps. Step 1, the differentiation of the separation zone. Step 2, the signals that induce abscission, such as environmental or developmental signals. And Step 3, signaling that results in the fruit separating from the tree. This photo is a close-up of a cross-section of the peduncle and abscission zone in a mature citrus fruit that was close to dropping. There are many biochemical and physiological processes happening in that abscission zone that will lead to the fruit to drop. Before the abscission zone forms, the vascular tissue is connected in order to supply the fruit with carbohydrates and water. But after the signals of abscission begin, whether they come from environmental stresses or just natural senescence, the abscission zone will start to form. The formation of the abscission zone includes physiological processes such as the synthesis of proteins, hydrolytic enzymes such as cellulase, which break down the cell walls in the abscission zone in order to start separating the fruit from the tree. After enough degradation of the cell walls happen and increases in sensitivity to ethylene production, the fruit will eventually detach from the stem. Abscission is a process that is heavily regulated by phytohormones, which are chemicals produced by plants that regulate their growth, development, reproductive processes, longevity, and even death. While there are many types of different phytohormones, there are 6 classes that are most abundant in major plant physiological processes such as gibberellins, auxins, ethylene, brassinosteroids, cytokinins, and abscisic acid. In the next part of this course, we will discuss which of these major classes are most involved in abscission that specifically pertains to pre-harvest fruit drop. The first and most involved hormone in fruit drop we will discuss is ethylene. Ethylene is a gaseous plant hormone involved in fruit senescence, abscission, and ripening. Citrus is classified as a non-climacteric fruit, meaning the fruit do not produce a large amount of ethylene for the ripening process as opposed to, say, fruits like bananas. When the fruit is removed from the tree, the maturation process halts, and although citrus is a non-climacteric fruit, the fruit does respond by dropping from the tree when treated with PGRs that induce ethylene production in the plant. Academic literature supports the general theory that pre-harvest fruit drop is a result of the increase in ethylene production, which ends in the fruit abscising from the tree. However, ethylene or other factors as a main cause of pre-harvest fruit drop in citrus is still not clearly understood. The stages of ethylene-induced abscission are very similar to the stages of abscission discussed earlier. Stage 1 is described as initiated by a stimulus such as natural senescence or external factors such as PGR treatments that would activate ethylene production. Stage 2 occurs when auxin in the abscission zone drops, protein degradation starts to occur, and the breakdown of chlorophyll begins, which all leads to the increased ethylene production. Stage 3 consists of synthesis of hydrolytic enzymes in the abscission zone that are responsible for cell wall breakdown, which leads to the fruit detaching from the tree. Stage 3 is thought to be when the fruit is beginning to respond to the increase in ethylene production. There has been copious amounts of research done on the link between ethylene and abscission for many crops, but for citrus specifically, there has been research to show that abscission can occur through an ethylene-independent mechanism, meaning causes other than ethylene result in pre-harvest fruit drop as well. A study done by Macejo in 2021 found that fruits that underwent a physiological abscission at AZC, shown on the top photo, produced higher rates of ethylene than fruit that were removed by force from the stem and not fully abscised, shown on the bottom. The findings from the study suggest that ethylene production does occur in the abscission zone, which would link pre-harvest fruit drop and ethylene production in citrus. Ethylene also works in tandem with other plant hormones that contribute to the process of pre-harvest fruit drop as well. Moving to the next major plant hormone involved in fruit drop, which is auxin. Auxin is a major class of plant hormone which plays major roles in many physiological processes such as growth and cell elongation. Auxin is found in all parts of a citrus tree, including abscission zone C, and its presence prevents the induction of abscission by ethylene. There are many types of auxins used in agriculture. Synthetic auxins such as 2,4-D, which is typically used as an herbicide, and NAA are used, as well as natural auxins like IAA and IBA. Numerous studies have shown that the application of auxin treatments can delay the pre-harvest fruit drop in citrus. Field test studies done by Augusti show that applications of 2,4-D prior to color break in Navelate orange, a variety that is considered highly susceptible to pre-harvest fruit drop, reduced fruit drop significantly depending on the dose applied. The higher dose tested, which was 50 milligrams applied, which is demonstrated by the green line, saw 11% less drop than the control, which is marked by the red line. Numerous studies have shown that the applications of auxin treatments can delay pre-harvest fruit drop in citrus. Applications of 2,4-D have been shown to reduce the expression of abscission-related genes in mature citrus fruit, which suggests that its presence is slowing the physiological processes of pre-harvest fruit drop mentioned in the previous sections of this course. As aging occurs in the fruit and leaves, endogenous auxin will start to decrease quickly, so it's possible that the resupply of auxin through exogenous applications prior to fruit drop signaling occurring may allow for the fruit to hang on the tree longer. The presence of auxin represses the ethylene production, so exploring the interaction between these two hormones gives a clearer picture into the mechanisms behind pre-harvest fruit drop. The interaction between ethylene and auxin in the mature fruit drop system has been studied in many crops such as tomato, which are climacteric fruits. However, whether this interaction is the primary mechanism behind pre-harvest fruit drop in citrus is still not understood. Since mature citrus abscission can be induced by ethylene treatments and delayed by auxin treatments, the interaction seems to play a large role in the pre-harvest fruit drop of citrus. It's important to note that some studies have shown that auxin applications can actually promote abscission when applied after the abscission process has started. It's thought that in citrus, the signals for fruit drop can happen much sooner than the fruit drop occurring. The stages of abscission in this model are very similar to the stages examined in the ethylene-only model. The process begins with signaling from either external factors such as PGR applications or environmental factors, or signaling from natural senescence, which is defined as the process of deterioration with age. As auxin decreases due to senescence, the ethylene starts to increase, and since the fruit is depleted of auxin, the sensitivity to ethylene will increase, and the physiological processes that are repressed by auxin will start to occur. The sensitivity to ethylene promotes the enzymes that degrade the cell walls, and finally, the fruit detaches from the stem. It is thought that applying exogenous auxin to the fruit will delay the sensitivity to the ethylene production. The next plant hormone we will examine will be abscisic acid, or ABA for short. ABA functions in many plant developmental processes such as bud and seed dormancy, fruit size, stomatal closure, and response to environmental stress. Despite its name, we know that ABA is not a direct cause of abscission in citrus, and this hormone is antagonistic to gibberellic acid. We also know that a relationship exists between color break in citrus and increases in ABA. Ethylene treatment, which we know induces abscission, has been shown to increase the ABA concentrations in citrus peel. Another mechanism that drives the accumulation of ABA is drought stress. Since ABA regulates stomatal closure, ABA accumulation during drought stress is a defense mechanism to close the stomata in order to reduce further water loss. ABA, although present in many organs, is known to accumulate in the roots during water deficit conditions. This accumulated ABA is then transported up to the canopy of the plant and then to the fruit, where the physiological responses occur that trigger abscission. It has been proposed that ABA's antagonistic relationship with gibberellic acid is another component behind pre-harvest fruit drop. Gibberellic acid, or GA for short, is a phytohormone involved in many plant processes such as cell division and elongation, flowering, fruit development, and seed germination. GA applications in citrus are well known for keeping the fruit green, but also applications have been shown to increase the tensile strength of the peel, which renders the fruit more resistant to any sort of physical damage, as well as having increased efficiency in the mechanical juice extraction process. GA applications have been historically used in citrus to delay peel senescence and extend the harvest window, since GA-treated fruit retain their green color and continue to photosynthesize. Applications of GA combined with 2,4-D at color break have been shown to be effective in reducing pre-harvest fruit drop in multiple varieties, including Valencia, Hamlin, Navel, and Clementine Mandarin. ABA and GA are antagonistic to each other. If you recall in the previous slides, ABA accumulates in the fruit during color break and is associated with increased fruit drop during drought stress conditions. One model that describes the relationship of these two hormones in regards to fruit drop states that citrus fruit always produce a baseline of ethylene that is not perceived by the plant until GA levels decrease during maturation or senescence, and the ABA increases as a result. When the ABA levels increase, that is when the fruit perceive the ethylene and respond by abscising. Let's do a quick summary. There are many plant hormones that are involved in plant processes such as growth, stress mitigation, and abscission. Ethylene seems to be the most involved in pre-harvest fruit drop according to the literature. Auxin has been shown to reduce pre-harvest fruit drop in citrus. As the plant ages, the endogenous auxin starts to decline and ethylene production increases, which is linked to fruit drop. Increases in ABA due to drought stress or senescence leaves the plant vulnerable to ethylene increases. And lastly, there is a relationship between the plant's endogenous GA and ABA levels and increased sensitivity to fruit drop. The next section of this video will summarize some environmental factors that contribute to pre-harvest fruit drop. As discussed in the ABA section of this video, drought conditions can trigger fruit drop. In citrus, ABA accumulates in the roots during drought stress and signals the plant to close the stomata to lessen any further water loss, which leads to increased fruit drop. The graph on the left is from a study published in 2013. It shows the sharp increase in ABA in the roots of citramella seedlings that were monitored under drought stress conditions. The top line is the average ABA levels of the plant in drought conditions, while the bottom line are the control plants that were continued to be watered regularly. In a matter of 5 hours, the ABA levels shot up in the roots, and after a short fall in ABA, they continued to rise for 30 hours. This data suggests that ABA accumulation can happen quite rapidly. Another environmental factor that causes severe fruit drop is freeze damage. While citrus is mildly cold tolerant depending on the variety, most varieties are susceptible to freeze damage. Freezing temperatures cause cell wall damage in all parts of the plant above ground. This cell wall damage can lead to increased ethylene production and therefore more fruit drop. The photo here was taken after the freezes hit Central Florida in February of 2022 in the Green Swamp region. There was extensive damage to the leaves, bark splitting, and substantial pre-harvest fruit drop. Flooding conditions can also lead to increased pre-harvest fruit drop. Citrus is a flood-sensitive plant with some rootstocks being more tolerant than others. Waterlogging creates anoxic conditions for the roots that starves them of oxygen, which leads to fibrous root damage, reduced photosynthesis, and overall stress to the tree. A stressed tree will try to balance the damage by dropping fruit in order to conserve resources for the rest of the tree that is being affected by the flood conditions, such as the roots. Now that we have gone over in depth how plant hormones and environmental factors play a role in pre-harvest fruit drop, let's talk a little about other factors that can contribute. Nutrient management is a critical part of proper grove management practices. According to Liebig's law of the minimum, as depicted by the graphic on the right, growth is dictated not by total resources available, but by the scarcest resource, meaning that if a nutrient management program lacks in one nutrient, the entire program could fail. Another phrase we can use is a chain is only as strong as its weakest link. Nutrient deficiencies induce a stress response and can lead to many negative side effects that will contribute to pre-harvest fruit drop. And since Florida has sandy soil and irrigation water that is high in bicarbonates, this already makes nutrient management a challenge. Sink strength is another factor that can contribute to pre-harvest fruit drop. Sink strength refers to an individual fruit's ability to obtain resources such as carbohydrates from the tree. The more resources obtained, the stronger the sink strength, as demonstrated by the fruit on the right of the animation. Although carbohydrate competition is associated with the June drop event in citrus, recent research has shown that smaller fruit in stage 3, or mature fruit that are close to the harvest period, tend to drop more than larger fruit. This is thought to be because smaller fruit have less sink strength and they stop growing. When the fruit stop growing, they will drop, and this can happen prematurely. Some field studies have shown that applying synthetic auxins such as 2,4-D to trees after the June drop has occurred can increase the sink strength of the fruit by increasing the size and carbohydrate content of the fruit. Now that we have examined a range of mechanisms and causes behind pre-harvest fruit drop, let's move on to how Huanglongbing, or HLB, contributes to these mechanisms. Florida growers have to produce fruit in an era where HLB is now endemic, so understanding these mechanisms and how the disease exacerbates them can help in making smart decisions in grove management practices in order to mitigate this problem. HLB is a bacterial disease of citrus that causes a vast amount of symptoms that leads to large yield losses and damage to the tree. The disease begins in the new flush, which is fed on by the Asian citrus psyllid. The bacteria is then transmitted from the mouthparts of the psyllid to the leaf, and the bacteria is then deposited into the phloem of the plant. As a defense mechanism, the tree recognizes the pathogen and then plugs the phloem as a method to stop the spread of infection, and as a result, no movement out of the leaf leads to photosynthate buildup, and the resources the tree needs to survive don't get transported to where they need to go, like the roots, and the roots will start to die back due to starvation. The fruit remain green, hence the other name for this disease, citrus greening. HLB's damage to the root system is a major concern in regards to pre-harvest fruit drop. HLB affected trees have been shown to have up to a 30% reduction in fibrous roots even before symptoms show in the canopy, while symptomatic trees have been found to have up to a 40% reduction in fibrous root mass. This reduction leads to less water and nutrient uptake capacity. As discussed in previous slides, drought stress conditions and nutrient deficiencies that can be induced by a damaged root system can lead to increased pre-harvest fruit drop. A reduced root system due to HLB can lead to drought stress response in the plant, which we know leads to a buildup of abscisic acid in the plant, which leads to increased pre-harvest fruit drop, as well as nutrient deficiencies, which renders the plant vulnerable to more stress response and then eventually leads to reduced yields. Recent research has shown that increased fruit drop rates in HLB-affected trees are correlated with the canopy density. Since HLB causes severe shoot dieback, a thinner canopy such as the tree on the right will have more fruit drop than the tree on the left, due to the fact that the tree just doesn't have the canopy to support the fruit. HLB causes a major disruption in the phytohormone system of a citrus tree. Studies have shown that gibberellins and cytokinins have been found to be repressed and ethylene-induced in HLB-affected trees. HLB-tolerant varieties such as Sugarbell have been found to have elevated levels of auxin and cytokinins, suggesting that hormonal profile and disease tolerance may be linked. There are some strategies that growers can use right now in their groves to mitigate the fruit drop related to HLB infection. Maintaining a well-balanced fertilizer program is critical to ensure that the tree has the proper nutrition to thrive. Controlled release fertilizers are a great way to deliver more nutrients over time to a damaged root system. This allows the roots to take up more nutrients before they leach away from Florida's sandy soil due to wet and humid climates. Fertigation can also be utilized, as this method has a very timely and cost-effective application while delivering nutrients directly to the root zone of the trees. This method can also be used to fill the gaps between granular applications. Overall, good management practices in the grove to control pests and fungal diseases will also be important in reducing the stress that HLB trees will endure. For more information about pest and fungal disease control, as well as fertilizer recommendations, please refer to the most recent Florida Citrus Production Guide. The use of PGRs, or plant growth regulators, which are agricultural grade products that contain plant hormones, to mitigate HLB symptoms in Florida groves is still a new and under-researched area. According to published literature, GA is able to increase the canopy density in HLB-affected trees. A study done on mature Valencias that received a monthly GA application using ProGib at 10 ounce per acre saw that the control trees lost about 8% of their density over the course of a year, while the GA trees maintained their density. Moreover, this study also found that the fruit yield increased by 30% on GA trees over the course of 4 years, as well as had larger fruit and less fruit drop. Applications of 2,4-D have been shown in recent research to reduce some fruit drop in HLB-affected groves. In a study done in 2021 on mature Valencia, GA and 2,4-D sprays were tested for fruit drop efficacy, and after one season of monthly applications starting in October and ending in December, the treatment that received both GA and 2,4-D had the best efficacy in reducing fruit drop, with 18% less fruit drop than control. Please refer to the Florida Citrus Production Guide for guidelines regarding the use of 2,4-D for fruit drop. Thank you for listening to the pre-harvest fruit drop section of the Flowering and Fruiting short course. For more information on the grove management practices that were discussed in this video, please refer to the latest Florida Citrus Production Guide, which can be found by scanning the QR code on the screen.